Today's notes are about a scientist named Gregor Mendel. Gregor Mendel is known as the father of genetics. He's from Australia, and he did a lot of work with pea plants to learn how traits are passed from parent to offspring. He was the first scientist to be able to do this effectively, which is why we often refer to him as the father of genetics. In his experiment, he worked with pea plants, and he did this because they were very readily available to him. You can see right here they reproduce in large numbers. They're very easy to work with, and new generations can come about in very short time. So he was able to have them reproduce very quickly. Before we begin talking about how Gregor Mendel used pea plants to help him learn about genetics, we need to understand a few vocab words first. We've talked about the word fertilization. This is when the male sex cell, such as the sperm, and the female sex cell, such as the egg, join together. Do you remember what that fancy word for sex cell is? When we have two sex cells or two gametes joined together, we have fertilization. The word pollination is just the fancy plant word for fertilization. So pollination is when we fertilize plants. So when the egg and the pollen is the sperm of plants, combine to make a seed. And there are two types of pollination for plants: self-pollination and cross-pollination. In self-pollination, we have the same plant fertilizes itself. Specifically, the same flower can pollinate itself. So you can see a flower has both female reproductive organ and male reproductive organs. The sperm, or the pollen, is created here and can rub against the stigma, which receives the pollen and then can grow the eggs below. So self-pollinate is when the same plant pollinates itself. Cross-pollination is when two different plants pollinate each other. And this doesn't necessarily happen that naturally, so it needs a little bit of help. Wind can blow pollen from one flower on one plant to another flower on a different plant. Or insects such as bees can take the pollen when it lands on the flower and then move it to another plant. And humans can also cross-pollinate plants. In Gregor Mendel's experiment, he used cross-pollination to make sure that he had control over which traits he was looking at. So when Mendel did his experiment, he studied one trait at a time, or one gene. For example, he looked at just height, or just seed color, or just the flower position. In pea plants, he learned that each gene, like height, has two different versions, tall and short, and we call those versions alleles. So it has two alleles, tall or short, when it comes to height. It's either going to be a tall plant or be a short plant. For seed color, the plant is either going to be yellow or green. It's going to be one of the two. When he did his experiment, I told you he did cross-pollination. So he took one plant and crossed it with another plant, or he fertilized another plant. So in order to do that, he actually cut and removed parts of a flower so that he had full power to pollinate and have control over which plants pollinate other plants. That way, he knew it was a very valid experiment, very accurate experiment. When he did his experiment, he started with two plants to study the gene of height. You can see he took one tall plant and one short plant and crossed them together. When he did that, he found that he had two tall plants. And he thought that was very interesting. So when he crossed the two tall plants from the first generation that he created 4 offspring and learned that he has 3 plants that are tall and 1 that is short, just like the original parent. And what that told us is that there must be 2 alleles. You can see big T, little t, little t. And that when you cross from the original, you pass 1 allele, so that this new generation would have 1 of each. And then when they cross again, they would send 1 of each of their different alleles. That would create 3 tall and 1 short. We're going to talk about these different capital letters later, but if there is 1 capital letter, it would be a tall plant, and if they're both lowercase letters, it would be a short plant. So what he learned is that these plants can pass their traits to their offspring. Now, when he was doing this experiment, he discovered a few things. The word homozygous, homo means same, is that there are plants that have the same alleles. For example, this tall plant has 2 tall alleles, whereas the short plant has 2 short alleles. But they're both the same. Big T is the same, tall, tall, and short, little t, the same. His first generation has to be heterozygous, meaning different alleles. 1 big T tall allele and 1 little t short allele, in order to create the offspring in the F2 generation. So after doing these experiments with all these different traits, he was able to discover 3 laws. Now remember, laws are statements that summarize scientific fact. We've talked a lot of this year about theories, and theories are accepted explanations based on the experiment. But what we learned was that theories can be changed, adjusted, added to anytime new information that we discover becomes available. For example, the cell theory, right? We started with 3 parts of the cell theory, and when we discovered more information, we added 3 more parts to the cell theory. A law does not change. His first law that he discovered is the law of dominance. And what this tells us is that a particular trait, such as height, can have 2 different types of alleles, dominant or recessive. Dominant alleles will always be expressed over the recessive allele. For example, big T, every capital T will be represented by a dominant, and recessive will be lowercase t's. And if you have 1 of each, the dominant will appear. So all 4 of these offspring will be tall. The only time you're going to have a recessive trait is if they're both lowercase t. The second law is the law of segregation. And what this one tells us is that, well, we know that every organism has 2 alleles or 2 versions of the gene, right? You might have a tall allele and you might have a short allele. But when we create the gametes, right here, these sex cells, we're going to split them up independently of one another. We're going to separate the alleles. And then the alleles will recombine once fertilization occurs, which is why we'll end up with the same number of DNA strands as the original. The third is the law of independent assortment, and what this tells us is that the genes are independently separated from one another during meiosis. So if you have a gene for height, and a gene for hair color, and a gene for eye color, and a gene for number of fingers, they're not going to be related to each other. It's not going to say if you have blonde hair, you'll also have blue eyes and 6 fingers. What it means is that they are independent from one another. So you can have a bigger range of differences of one another. So you can see you could maybe have blonde hair, brown eyes, and 5 fingers, or blonde hair, blue eyes, and 5 fingers, or blonde hair, blue eyes, and 7 fingers. What this does is it creates more variations, more differences between our organisms, which is going to help us survive in tough situations. Please write your summary and questions.